Good morning, everyone. Good morning, students. Today we are going to discuss a topic called as clinical trials, right? What do we know about clinical trials, right? And why it is important to learn about clinical trials? Is it that necessary? We have to learn clinical trials, right? And what is the importance of clinical trials? We'll be seeing in today's class. And nowadays, whenever you see from the starting of the pandemic, we have been hearing the research is happening for to cure the COVID-19. Our vaccines are being developed, and the clinical trials are happening by different countries and by different companies. And some companies and some countries says they're in the final stage of developing the trials for developing the vaccine for clinical trials. And some companies says, some countries says they already have one vaccine, they're going for second vaccines. So now let's have a look into it. What generally happen in clinical trials? What exactly a clinical trial is, right? Next, generally when you see, it is a big question mark for the most of the people. What is a clinical trials and what exactly will happen in there? So it's as simple as you know it. It's not that difficult. It's as simple as you know it, ma. Let's discuss today, right? So now, in next slide, we'll be seeing the entire process of clinical research, like from the starting of drug development to end of uh, the drug into the market, entry of the drug into the market. Right? When you see this, okay, when you see this image, it will specifically says from the starting of the drug development, like means once you have the drug with you, okay, from preclinical development till the drug being used in the market. Okay, so let's see here. Preclinical testing, like we already seen in preclinical testing, we'll develop a molecule, we'll write a protocol for that molecule, okay, and that one will be used in animals. That one will be used in animals like rodents, non-rodents, like we have seen in pharmacological approach and toxicological approach. We'll be using this in animals like rodents, non-rodents, rats, mice, and certain animals, bigger animals like monkeys, dogs, or like small animals like guinea pigs. Everything they'll be doing clinical research in that animals, and we'll be getting a data like which one is safe dose, which one is toxic dose. Does this drug really have a efficacy or not? Okay. And next phase is clinical research itself. Like in here, you'll be doing the dose finding in phase one of clinical trials, and in phase two, and apart from dose finding, we'll be seeing the safety of the drug. And in phase two, we'll be seeing the efficacy. We'll be seeing the efficacy. See, the safety of the drug is important, so we'll be seeing the safety of the drug in phase one. And phase two, we'll be seeing the efficacy of the drug. Does this drug really having an efficacy? Does this drug is really having an appropriate therapeutic efficacy towards a specific disease condition or towards a specific target identification or target binding or not? So once that thing is done, then we'll go for phase three, right? So here we are going to see like what you call as confirmatory phase. Like, so phase one is safety phase, human pharmacology phase. Phase two is therapeutic identification phase, and phase three is confirmatory phase. Means here we'll be seeing the drug really have a therapeutic efficacy, and the drug is really have a toxic effect. So on which group of people this one is effective, on which group of people this is not effective. If you can see here, there's a comparative efficacy, comparison of new with the standard therapy. Supposing if there is RC, when you see NSAIDs or antibiotics, we have a number of antibiotics. Means, right? All the antibiotics didn't come into at a time in the market overnight. Every drug has a different entry level in the market, right? So when you have produced different, take example of your ciprofloxacin, cephalosporins, class 1, generation 1, generation 2, generation 3, generation 4, generation 5 also, right? So what happened, each phase of each generation of cephalosporins is having specific effectiveness, specific drawbacks, right? So here, when it is gram phase generation 1, generation 2, generation 3, so some are active against gram positive, some are active against gram negative, some are active against both gram positive, gram negative, right? So here, we'll be seeing the benefits, okay, Or will be comparing the phase 1 of ciprofloxacin with phase 2 and phase 2 with phase 3. They'll be seeing, so which one is better? Whether which supposing they already phase 3 phase of 3 generations of cephalins were available. So when you're developing phase 4th generation ciprofloxacin, sorry, cephalosporins, what happens? You are going to check whether this generation 1, generation 2, generation 3 of cephalosporins are active, or the new drug whichever is going to be in generation 4 is active or better version of it. So they'll be comparing it with a new drug with a standard. That means already drug is there in the market for this. As your drug is going to make a better improvement in the market, better improvement for the patient, right? So you'll generate a data, you'll go for data analysis, you'll go for data analysis, okay? Next, then when you see it is safe and properly used in humans, then you will be getting a testing approval from the government authorities. Then the drug will be tested effectively, right? This is, and like if you see these things again here, phase 1 will bring 20 to 100 people, 20 to 80 or 100 people, healthy volunteers, and phase 2 in hundreds means from 100 to 300 to 500, and in phase 3 in thousands means from 800 to 1000 or 2000 to 5000, like that. Okay, and if in phase 4 we don't have any restrictions like that, because phase 4 is nothing but your post marketing surveillance, which will be happening, which will be done in wider population. Next one, see whatever we are going to discuss, everything is in here, like clinical trials. What exactly is a clinical trials? What exactly is a clinical trials? So all these things, all the terms, okay, like when you see scientific comparison, behavioral studies, treatments, safety, vaccines, devices, factors, medicals. So each and everything will have a link towards your clinical trials that we'll be seeing in the upcoming slides. How and what, okay? So what is clinical trial? We have different definitions from clinical trials. So we'll be seeing our 2 to 3 definitions, okay, right? So CT means clinical trials here, okay? So some definition from S.K. Gupta says, clinical trial is a structured process means we are going to create a structure, how we are going to do it. What is the step 1, what is step 2, what is step 3, what is step 4? How, if there is any drawbacks, if there is any hurdles or any dangerous or any unwanted unknown reactions, if anything happen, how we are going to overcome it. If there any problem happens, how we are going to overcome it. Everything will be written, whichever is known to the human being and knowledge, everything will be written. Because at this level we don't know 100% of the drug, we'll be knowing just a 5 to 5% of 10% of the drug, so remaining 90% we have to find out. Okay, so it is a structured process by which new pharmaceutical medicines, it can be a pharmaceutical medicine, it can be a medical device, it can be a biological active agents. Okay, so anything which are evaluated for these 3 parameters, safety, efficacy, and effectiveness. They'll be doing to see safety, efficacy, and effectiveness, okay, towards the humans, or these drugs are safe and effective, and show its effectiveness towards the disease conditions or towards the treatment which you are going to give. Okay, next one is some other definition. A clinical trial is defined as a systemic and prospective. So it's a systematical way, a structured approach. And prospective, why we are saying prospective means, it's a prospective study means we are planning to do it. How? So when we say we have prospective, concurrent, and retrospective. Prospective means in the future how you are going to do, you are going to plan. Concurrent means when it's happening in between you are intervening. Retrospective means the thing is happened, now we are examining it. Okay, so prospective means to the future, or in the future how you want to do it. Okay, so clinical trials can be defined as a systemic, prospective study aim to compare the effect, the value of interventions against control group in human subjects. Remember, ma, why we have a new term control group here means whichever maybe the clinical trials happen, we'll be having one test group, we'll be having one test group, and we'll be having one control group. So we'll be seeing the effects in these two groups, test group and its control group, or we can say standard group versus test group. Okay. Next, another definition. This one was given by ICHGCPMA. ICHGCP is nothing but International Conference or Harmonizations, and good clinical practice is one of the high-end organization we can say, which will look into the clinical trials happen. Right. So clinical trials or study is defined by ICHGCP as beginning any investigation in human subjects. Okay, any investigation in human subjects. Investigation means any type of works, any type of things which are you are doing, and you are involving human beings in it. Okay, so remember I am saying human beings, not living beings. Okay, see why I'm saying living being means, see we have preclinical trials also. In preclinical trials, we'll use animals. Okay, those won't come into human beings. That's what I'm saying. So this is a definition of clinical trials means we are using in clinical trials, we'll use only humans, right? So that's why we're saying any investigation in human subjects intended to discover or verify means which you are going to discover. If you are going to discover new, or you are going to verify it. Okay, you're going to discover the activity of the drug, or you are going to verify does this target is or this that this gene is responsible for this disease condition. Okay, we are going to use human subjects to discover, to verify the clinical pharmacology or other pharmacodynamic or pharmacokinetic effects of an investigational product. Okay, it means we are going to use investigational drugs in this. Okay, and to identify any adverse reactions are there in a given active pharmaceutical ingredient. You are going to verify that one, and you are going to study the ADME of your investigational drug. Okay, with an object of ascertaining safety and efficacy means. So the entire theme says you are going to use a new drug. Okay, and to find out the effects, to find the safety and effectiveness of the drug, you are going to use humans to find out the effectiveness and safety of the drug. If you are using humans, that is called as a clinical trials. Okay, so how can we say the drug is safe and effective? Right, we should have certain objectives to find out. Okay, so even if we use humans, we should know certain parameters which should be identified. Like we are going to identify clinical pharmacology, we are going to identify pharmacodynamic effects, we are going to identify adverse drug reactions, we are going to find out ADME of the drugs. Okay, so like this, we have a number of things which will be involving in one clinical trials. Okay, hope the definition is clear. Okay, see, these are the first known clinical trials which are recorded. We already seen this one, James and Lind. So these are the first people whose clinical trials has been recorded in the histories. Maybe these things, which there might be so many things which has been happened. We already discussed in the introduction of your clinical research. Okay, so one of the notable thing which are clinical trials which have happened in history is a scientist named James Lind, who is a seaman. Okay, where he has conducted a trial in 1747. Okay, as a treatment for scurvy. These are the images which shows. Okay, okay. So which will happen in a ship actually, which happened in a ship actually. If you see in histories, okay, so earliest evidence of clinical trials in 20th century. Okay, so in 20th May, 1747, James Lind, what he has done. So at that time, there was no standard treatment for scurvy to treat the patient. So what happened? Being a scientist, being a doctor, this James Lind, what he has done. So people who are affecting with the scurvy in a ship. Okay, so what he has done, he has divided people into two groups. Okay, so to one group he has just not given anything, no medicine, nothing. To other group he has not given medicine. What he has done, he has used lemon and oranges. He has given a juice of, if you see this bag, just say like a color of yellow or green or orange. So he used lemon and oranges. So those group of people has recovered so fast in 6 days. And this was the first known and recorded clinical trials in history. Okay, so where in which to one group of people he has not given any medicine, to another group he has just given not medicine, he has given a dietary supplements like lemon juice and oranges. Okay, where in which they recovered quickly after 6 days of giving this lemon and oranges. So one of the well-known and recorded clinical trials on 20th May 1747 in a ship. Okay, and going back into the history, next thing like randomization. So I think you already know about randomization, right? So the randomization was first time used in clinical trials, introduced in clinical trials by Fisher, scientist, in 1926, and he was an agriculture researcher. Next, okay, next one is Amberson et al. So the group of scientists, one of the major scientists was Anderson. Okay, so 1931, the first clinical trial that used a form of random assignment of subject to study group and control group. Okay, so randomization of first used in by the scientists in agriculture research. Okay, and in clinical trials, Amberson et al. Are the first scientists to use random assigning of subjects in a study group, like for control group and test group. Like I said, in a clinical trials will happen in two groups: control group and test group, or standard group and test group. Okay, okay. And next, known one of the known trials which are recorded here is, so like here we said a flip. They randomly use the people into standard group and test group. And next thing here is, it was actually a treatment trials for your pulmonary tuberculosis, right? So here how they determine, a flipper coin determines which group of 12 subjects would receive sanocrosine, a coded compound for tuberculosis versus control in that period of time in ages in 1930s and all. So here what happened? They divide the people with the help of random assigning or randomization technology. But which group to receive standard treatment and which group to receive control treatment? So this was designed just by flipping a coin, just by flipping a coin. Okay, going back to the next one. Yeah. So British Medical Research Council in the year 1948 for the first time used properly randomized control for streptomycin, the treatment of pulmonary tuberculosis. And dated back one of the best thing like principles of blindness. Like you have single blind, double blind, and triple blind studies. Okay, so that was the first introduced by Amberson. So you can see in previous slide we have seen Amberson was et al. Was a group of people in 1931 who has randomly assigned the subjects into control group and test group. And here, same Amberson was the person who has used the principles of blindness. So what is the principle of blindness? That will be in upcoming slides. Okay. Next one in 1938 by Dechi et al. Who give a saline solution to subjects in the control placebo group in a cold vaccine type. So what they have done, they have used they have done a vaccination trials where in the control group they just given a placebo. Means I think you already know about placebo. It's an inert molecule. We don't have any therapeutic effects with it. Okay. One group with that placebo, and another group will be having a medicine in a vaccination trial. Next thing, yeah. So if we see the clinical trials have been evolved in a much faster rate and much higher rate after your Second World War. Okay, the clinical trials has evolved into standard procedures and a much higher rate. Okay, after the Second World War. So why this thing happened means in the Second World War there is a huge loss for all the countries, the winning countries and the losing countries. And when there was a huge biological warfare which has been used, so there was a new diseases which were unknown. The new diseases and new pandemic conditions happened because of the change in the climatic temperatures, where we have used atom bombs and so many warfare, so many gunpowders and so many things we have used. So what happened? There is sudden change in the people's health, where which has lead to change in the environment, change in people living pattern, which started developing new disease conditions and all. So to treat all those new disease conditions and all, so people started developing a new drugs, new clinical trials. Okay, right. And next one. So which includes the use of, as I already said, it has become a standard procedure after Second World War. So what are the new things what they started after Second World War is they started using of control group and test groups. Okay, they started random allocation of patients in control group and experimental control group and experimental group or control or test group, and I used blind and masked assessments. Okay, so these are the new things which have been evolved after Second World War. Okay, so that was the thing. So here, what are the basic principles of any clinical trials, right? So whenever you're doing a clinical trial, you should follow certain basic principles and basic things. Like clinical trials must employ one or more intervention techniques. That means whenever you're doing clinical trials, any one of these following things should be there in clinical trials. Like first one, prophylactic means whichever drugs, whichever devices, whichever vaccines you are going to use are those for prophylactic drug control or disease control. Recently, when we do any surgery, we are going to perform certain pre-operational procedures like giving prophylactic treatments, like giving like a nil by mouth or TT or certain drugs for lidocaine test dose or certain antibiotic test dose we will be giving. So those are for prophylactic use. So is your drug comes under this category, or is your disease which are you are going is your device which are you are going to use for diagnostic of any disease conditions. And next, therapeutic device like for radiation therapies and all, or robotic surgeries and all, right? Nowadays, when you see robotic surgery, they are saying robotic surgery is one of the better, good invention in the medical field. So even to use those machinery, when to do robotic surgery, the procedure need to be undergone clinical trials. When the device need to be undergone clinical trials, then only they will get approval. Next are regimens. The things which are developed in the clinical trials are being a regimens. Like whenever you see regimens, one of take from your subject like therapeutics, you will be having an peptic ulcer disease or H. pylori induced peptic ulcer. You have 3 drug regimen or 4 drug regimen. From where will get those regimens, or this drug has to be used in these disease conditions? How come we came about it? Because those regimens have been used in certain group of people in clinical trials. Then we came across this thing. And next one are the procedures. Like whatever with the suppose thing, whenever you want to do certain clinical trials, whenever you want to do certain diagnostic procedures, or whenever you want to do certain surgeries, okay. So there will be a certain procedures which has to be followed, right? So those procedures has to be undergone certain clinical trials and tests. So those things will come under your basic principles of clinical trials. Means whenever you are conducting any clinical trials, any one of those things should be there. So let your clinical trials be on a drug, on a device, on a surgical procedures, It can be a regimen of the disease conditions. It can be anything. Like regimens in the sense, take a present example, pandemic example, like COVID. Like previously, we didn't have any regimens or drugs. We don't know. First, this hydroxychloroquine. Now they're giving hydroxychloroquine. Along with it, they are giving certain other azithromycin. They are giving paracetamol. They are giving certain vitamin tablets, like vitamin C, vitamin D3, multivitamin tablets. So this is a regimen to treat COVID. So how they came to know about this? They had done certain clinical trials, and they came to know this is a regimen, a better regimen to treat the clinical to treat the COVID-19. And other important thing you have to know all the drugs, whatever they say, right? It may not show 100% efficacy in all the patients. They don't show efficacy in all. So whenever you're doing clinical trials, any one of these things should be there as a intervention or as a basic principle in your clinical trials. Okay. Okay. So without active intervention, a trial is an observational one. Means where you just see what kind of treatment they're following. Okay. And we'll just follow it, and we'll just come to a conclusion. What is it and all? Okay. Going to the next one. Right. Yeah. So to compare the interventional group, a clinical trial must contain a control group. Means like it will be like a standard group and control group, right? You'll be having to one group, you'll be giving a standard treatment which will already existed in the market, and to another group you'll be giving a treatment of your drug. Okay. Okay, next, it is essential that the control group must be sufficiently homogeneous with respect to intervention group. Means what kind of subjects you are going to take in intervention group, same kind of subjects should be taken in control group. Or suppose consider uh, suppose if you are taking a healthy volunteers in control group, you should take healthy volunteers in interventional group also. But instead of that, if you take healthy volunteers in control group and unhealthy people in interventional group, what happens? It leads to an bias. It leads to a bias where you don't be seeing the exact therapeutic intervention which you wanted to see, which leads to a problem afterwards. Okay. Next thing is the new interventions can be like new intervention can be compared with current standard therapy. Supposing previously we didn't have ceftriaxone to treat the so many disease conditions in broad spectrum antibiotics, but we have some other antibiotics like certain first generation of cephalosporins and second generation of cephalosporins, right? But we came with a new generation three ceftriaxone, which is almost widely and broadly used antibiotic in the era, okay, like that. Okay, so clinical trials, the investigator cannot dictate the individual participants to do as per the requirement, unlike animal studies, right? So this point clearly says any researcher or any investigator is nothing but a person who conducts the clinical trial. He cannot dictate a subject who is part of clinical trials to do like this, to do like that, just like a dictator rule or those kind of things. Those things cannot happen. Here, the subjects are having 100% individuality. If they want to be a part, they'll be a part. If they don't want to be a part, they can leave at any time of clinical trials, even at the final stage of clinical trials also. If they feel they're not being good, there are certain other issues which they want to leave, they can leave it. They can leave from the site. They can leave from the site of the clinical trials, no problem. Yeah, but certain minor terms and conditions will be there. If he wantedly, willingly need to uh, go out of the study without any proper reason and all, then he will be having certain things need to be taken care and all. That we'll be seeing in the upcoming slides. Okay. Whereas living from the clinical trials won't be happen in case of preclinical, because in preclinical we'll be using animals where we it's almost like we are just dictating the animals. We are sacrificing them. We are giving them pain. We are giving them unnecessary things. We are making them feel bad. We are making them to get disease conditions and all. Because whenever it's with the clinical trials, to certain extent it's fine. But when specifically speak about chronic conditions like your cancer, what we will do, we will induce the tumor inducing gene in rats, We let it the tumor grows in the rats or mice, then we'll start giving treatment to it, right? So here we are inducing the disease, we are inducing the treatment, where we are asking them, making them to suffer and all. So like this, uh, in a healthy animals, what we have done as a dictator rule in preclinical trials, thing won't happen in clinical trials. Okay. Next, the design and conduct of any clinical trials require three important considerations. Okay. First thing, study must examine valuable and important biomedical research question. So your clinical trials must be having capacity to examine the important. Suppose thing, your thing was, can your title of your project was, can ranitidine cause any tumor growth, or can ranitidine increase the chance of getting tumor growth? If that was your question, your protocol and your research study should have and that much capacity to find out whether it's really having the capacity to cause tumor or increase the chance of getting tumor. This thing should be there. Next, it must be designed based upon a rigorous methodology and can answer your basic specific research question. Means, so whenever you are using or applying any research methodologies in any of your clinical trials, that should be specific. Like give an example, like whenever I said, if you want to use, if you are conducting clinical trials or any research project where you are collecting the same data from same people at two different point of times. So here there is a link. The link is same group of people and same questions. There is a link. So at that time we'll be using paired t-test, right? There is a link. That is one of the methodology which we'll use. So when we take this kind of things, we will be seeing the quality of life improvement after your interventions, right? And when if you are not collecting the data from same people at two different points of time, so what happened at that time? You can use unpaired t-test or single t-test, right? So those kind of things. Your research methodology which you are going to implement, that should be done after a thorough research survey, and that research survey should answer your question. Should answer your questions. Next, must be based on set of strong ethical principles where you have to follow all the principles. Like we have so many things, like Belmont reports, Declaration of Helsinki, Nuremberg codes, and so many things here, which we'll see in later classes. So many ethical principles are there which we have to follow. Okay, and next one is minimize the risk to trial participants. See, minimize the risk to trial participant means we already know the people are at risk. People will be at risk when the clinical trials will happen. But the major thing which what we have to assure is people are not exposed to unnecessary risk. Unnecessary risk. So what I'm saying is at this phases, at initial phase of clinical trials, we don't know the entire side effects or risk of the drug. Okay, but if the drug is from a cancer, anti-cancer drug class, we already know there are so many their side effects and IADRs, right? So being voluntarily known, we cannot take healthy volunteers in this type of trials. So generally, for cancer, like generally when we see phase one, in phase one we'll be taking healthy volunteers, right? But in cancer, we don't be having phase one specifically. So what we don't want to take healthy volunteers and we will give the anti-cancer drug to them being a known adverse drug reactions of that class of drugs. We already know it will have nausea will be there, vomiting will be there, and alopecia will be there, and etc. etc. We already know it. So even after knowing the toxicity of the drug, we don't want to give or we don't want to disturb the health of the healthy volunteers. So what we'll do at that time is we'll be doing phase one in diseased patient means who are already having cancer. Okay. And going to next slide. Okay. The choice of an appropriate study design based upon the following factors and consideration means whenever you are choosing any study designs, like I said, if you are going to collect the same data from same group of people at two different points, you'll be taking any paid statistical methods like paired t-test. Okay, so like that we have to see these considerations. Okay. Yeah, it's almost similar like basic principles. The first thing is the ability of the study design to answer the primary research question. Okay, your study design can answer the research question appropriately or not. Okay, next, whether the trials is studying a potential new treatment for a condition which an established effective treatment is already exist means uh, consider we already have treatment for cancer or tuberculosis or some other disease conditions. Are you going to provide a new drug for the same disease condition that plays a role? Again, that factor should be considered. Next, whether the disease for which a new treatment is sought is severe or for life threatening. So is your treatment or your drug which you are producing or the device which you are preparing or the procedure which you are creating is to save a life of a severely ill patient or critically ill patients or life threatening patients. So these are the factors which you have to see when you are designing a study appropriately or selecting or choosing a study appropriately. Next, the choice of the same thing. The other considerations like the probability and magnitude of risk to participants. Next, next thing comes to benefit to the patients, the probability and magnitude of likely benefits to the patients. Next, the population to be studied, its size, its availability, and its accessibility. Everything should be checked. Now we'll discuss the parameters one by one. So your study design, if you say, like you have seen power of the study or something you have studied in your something like you have studied in last year by statistics, like an appropriate study design must include sufficient sample size. See, sufficient sample size means like whenever you see a pilot study, maybe that's enough if you started, if you take people from 10s or 20s or 30 or 50 for entire study. But when you see a clinical trials, you need a members like in hundreds that should be taken care. It means appropriate people should be there whenever you're for using any study design. Like statistical power, like the p-value and all that should be taken care here. Next, you should have a proper control on biasma. So this is an uninvitable and unavoidable things which will happen in almost all the clinical trials, which should be taken care properly. So to avoid this bias, we can use randomized clinical trials or crossover or cross-section trials. Okay. Next, the types of clinical trials. So what are the types of clinical trials?